Hi, my name is Shari Wiseman. I'm the chief editor of Nature Neuroscience, and I'm here today with Claire Magnon, who is at the INSERM. And Claire, to begin, can you tell us a little bit about the field of cancer neuroscience and how you got interested in it?
Actually, at the beginning, I was working in the tumor microenvironment field. I didn't work at all on neuroscience. I'm an oncologist by my education background, and I was starting to be interested when I finished my PhD. I was working on angiogenesis, and I was thinking that maybe there is nerves everywhere in any organs to control them, and I was thinking, why not? Is there any innovation in tumors? So that was the first time I've got this idea. And then I submitted this idea to a different PI in the United States, and they were very excited, and one of them said, yeah, come and start in my lab. This was Paul Freinet.
Ah, I see. And so what kind of work did you do in that lab?
So in that lab, so the project was to first time find if there was nerve fibers in tumors, and if so, we wanted to know if those fibers had a key role in tumor growth and cancer cell dissemination. The first question was whether these nerves had a key role in the dissemination of cancer cells. But the first experiment I did was to denervate tumors chemically and surgically, and by cutting nerves, and I couldn't get any engraftment of human cancer cells. So our first question, we couldn't respond to it because we didn't have any tumors. So we had to look at tumor growth, tumor initiation, and we found that the nervous system, and particularly the adrenergic nervous system, could control the early stages of tumor development.
Interesting. And so what kind of work has your lab been doing more recently?
So step by step, we found that the brain was truly involved in tumor growth, not central tumors, but peripheral tumors. So it was kind of exciting uncovery. And now we are going to work more and more in that direction. And trying to find out which kind of nerves or molecules can be involved in the interplay between the central nervous system and peripheral tumor growth.
Interesting. So this question came up in your talk, and I think it's a really interesting one, which is why sort of from an evolutionary standpoint, we always like to think that whatever exists in biology exists for a reason. Why do you think, how do you think this process arose? Why does your body, grow nerves to feed the tumors? Is it just sort of an accident? Is it sort of a repurposing of a useful function?
I think it's a repurposing of useful function, like you said. Tumor, I like to say that a tumor is a smart little micro organ in the organ, and the tumor can play with many population of cells that are infiltrating any physiologic organ, and attract and corrupt all this cell population for its own fate. This is.
So in your talk you mentioned that you've been studying neurogenesis within the tumors. Can you talk a little bit about those findings?
Yeah. So actually at the beginning we found there is axonogenesis, so neurite outgrowth from pre-existing nerves, and then we moved forward neurogenesis, tumor neurogenesis, based on progenitor cells, neural progenitor cells that we found in prostate tumor tissue, but we found them also in breast cancer tissue. And those cells, at the beginning, I had not the idea to find them coming from the brain. We thought that maybe those cells were coming from the neural crest, and they were sitting there. But it was unexpected result to see that those cells were coming from the brain and from the neurogenic niche, one of the neurogenic niche, not both of them. And we found functional data and found that those cells could differentiate in adrenergic neurons inside the tumor.
I mean, this to me is just wild, you know, and cancer biology, I think a lot of it I find wild, but that neuroprogenitors can migrate somehow from the brain through the vasculature into a tumor, become adrenergic cells, and feed the tumor. I mean, that is just remarkable.
Yeah, to be totally honest, I was very skeptical at the beginning, and when I find the first result I could see, I thought that was an artifact, and we had to reproduce the data more and more to be convinced. And one day we had to, we must, and we moved forward and we did another experiment to stain neural stem and progenitor cells in the brain to see if those cells would be the ones that we could see in the tumors. Otherwise, we couldn't make any conclusion. And it was the case. We find them running in the circulation and going towards the tumors, and there is a specific tropism towards the tumor.
That's incredible.
Yeah.
And those cells promote tumor growth also?
They promote tumor growth, early stages. As I said, since they could differentiate into adrenergic nerves, and we know that adrenergic nerves are involved in the early stages, it's totally similar to what we found previously in our first publication in Science.
So what are kind of the translational implications of what you're finding? How can these how can this be used to treat cancer, I guess?
We have a lot of expectation in terms of translation to the clinic, and so there is some epidemiological data that shows blocking the beta-adrenergic signaling with non-selective beta blockers, very old molecules, very well known to treat cardiovascular disease, and those molecules can have an effect on tumor growth, survival, recurrence, and also decreasing the risk of metastasis. But what we would like to design now is to have new molecules targeting specific beta-adrenergic receptors. The old molecules target part of the receptor, but not all of them, and target more the beta-1 adrenergic receptor, which is expressed in the heart. We would like to have molecules that do not touch and target the heart, but only the tumors. So targeting beta-2 and beta-3 adrenergic receptors.
Oh. And would you give those drugs systemically, or you would inject them into the tumor?
We would like to have pills. We would like to have a systemic administration. And since we have seen that beta-2 and beta-3 adrenergic receptors are very strictly expressed in tumor tissue, we expect not to have too much side effects. We have to make more experiment and design and to make sure that we have a selective effect on the tumors.
Oh, very interesting. And I guess what other kinds of directions is your lab going in from here? What are some other things you guys are working on?
Actually, we have two branches. We have one, so like I said, it's to look at the crosstalk between the brain and the tumor. But the other branch is to look at the interconnection, the crosstalk between nerves within the tumor tissue and the other component of the tumor, such as the immune cells. And I think it's a very interesting and exciting field now.
Do you want to talk maybe about some other work that's going on in the field that you think is really exciting or really interesting?
So the field has started on peripheral tumor, and now there is more work in central tumor as well. So it's very interesting to see the connection we can have between neuroscientists working in high-grade glioma and oncologists to support that field and going forward in different direction. And immunologists, not only the ones that used to work in cancer immunology, but all kind of immunologists, and they are more interested now in cancer, and we can, collaborate all over the world.
Yeah.
Even scientists coming from the artificial intelligence aussi are really interested in the field. So we can manage new techniques, new collaboration, and share ideas. And I think it's growing field. So people coming more and more and feed the field in a very interesting way.
Oh, very interesting. What kinds of applications of AI technology are you envisioning?
Right now, maybe I can tell it's too early to describe the thing.
Okay.
But we start to talk about this.
I see. Okay. Well, that's great. All right. Well, I think we can wrap up there. It was great chatting today. Thank you for joining me.
Thank you very much for your question.
Yeah.